High-voltage electron microscopy allows three-dimensional imaging of a segment of this insulin-secreting pancreatic cell. Relatively thick slices of the cell are viewed in the microscope from different angles, which allows us to reconstruct a three-dimensional image. Stepping through the image from the top reveals the complexity of cell structure. Focusing on the Golgi apparatus, individual membranes can be traced, and we can appreciate the size and shape of various compartments. Using these outlines, a computer can construct a three-dimensional model of the entire segment. Here we see the stacks of the Golgi apparatus, each traced in a different color. The cis-Golgi, where proteins are first delivered to the organelle, is light blue, and the trans-Golgi network, where they exit, is red. Shown in dark blue are the secretory vesicles into which insulin gets packaged after leaving the trans-Golgi network. Many little transport vesicles, shown in white, surround the Golgi apparatus. They transport cargo between the cisternae or back to the endoplasmic reticulum. When all the other organelles are combined into a single image, we can see the incredible crowding of organelles in the cytosol. Here, mitochondria and microtubules are colored green. Endoplasmic reticulum and ribosomes are shown in yellow. The purple organelles are probably endosomes. Given this apparent clutter, one cannot help but wonder how all these components work in synchrony to allow the cell to achieve its tasks.